Okay, so it's 2 o'clock, so let's start off the class today. If you can hear me, you can shake your hands, raise the hand, or shout out, I can hear your voice, something that would be great. Okay, that's good. At least one person or a few can hear my voice. Just before the class, bit of announcement for the this week and next week schedule. So we're going to learn the topics of the heritability today, and that will be the last session of last topic of the session 2. And we are going to have the review session on next Monday and Q&A as well. And review session will be quite short. It depends on how you ask the question. I mean, I'm not going to cover all of the things on the review session. So it's mostly focused on the questions or the comments from you guys. So that will be on Monday. And also the next Monday, we're going to have little practice for the exam, because the exam will be online and it will happen on Blackboard. And I'm trying to make sort of the system everyone can take the exam securely without any unexpected situation. So we are all going to log into the Blackboard system and try to do some mini, practice for the your exam on Wednesday. So if you want to, know about the format of the exam, Monday is your time, and also be prepared that situation on Monday too. So there is no excuse for not having exam on Wednesday due to your systematic problem, right? So I'm not accepting this things, because we're going to have that practice on Monday, right? So that's the important notice. And exam will be on Wednesday. So again, if you are too nervous about that preparation, leave your comments, can come to Monday session and prepare your exam. Okay, so that's the good thing. And then another announcement is where I'm going, Slack. So if you look at our Slack, I simultaneously update our lecture video on general channel. So you can click on here and you can go to, you can have the previous session on YouTube anytime you want. And there's no, the barrier onto this video watching, right? So, I mean, I'm not sure how many people have that, YouTube video for your study, but given the number of views so far, it's about 5 or 6 per video. So it's great number. So hopefully many students will prepare before the class, because otherwise that will be quite tricky in the next session. So I'm open all of the research for your learning. So I mean, I don't want to get sort of the feedback on the insufficient learning research for your preparation. So hopefully have your chance, increase your probability having good, exam scores. So I'm all open to your request, not the questions just before the exam. So that would be quite bad solution for that. Okay, so so related to that, so leave your questions as far as you can before the Monday's class, right? So after the Monday class, there is very little chance to have your question feedback on after Monday class or before the exam. So I mean, given the previous years and previous semesters, some students rushing out the questions just before the exam, and that's not going to work anyway, right? And also I have another schedule for my work and research, so I'm not taking all of that just before the exam. So don't take that the ridiculous way. So please be prepared at least this week or weekend. I'm live, so or Monday class, so that then you can, check your understanding before the exam. Okay, so last topic today is the heritability, and this is the sort of the conclusion remark of how people see the inheritance in early times, right? So this is the continuing discussion from the biometrician, and especially from the Francis Galton and Charles Darwin, and how people see the continuous traits and how they measure that characteristics in the human traits. And this heritability topic is the critical things in modern genomic research, especially in the polygeny research and all of whatever the biomedical stuff. And heritability stuff probably I'm the one of few classes teaching this topic in our country for the genetics topic. So you will have that heritability concept in the context of the biomedical research and also the human disease genetics. So have it on yourself, and also be lots of discussion with me. That would be great. Okay, so learning objectives and aims. So the first thing is the understanding the quantitative traits in natural population and the terms describe the normal distribution of the human traits. And also we're going to discuss about how we measure the genetic similarity. And also we're going to discuss about the components explaining the phenotypic variance, right? And lastly, we are discussing the concept of the heritability. And today's topic, we don't have the good textbook for that. We actually have the good textbook for the heritability stuff. That's written by Benjamin Neal, and that's up here on the Amazon. So you can get an ebook from there. But I think that's at least the intermediate level. And also the topics today is quite beginner level. So I'm not going to the details on that, but just for the introductory stuff. So I mainly put some learning materials from the short articles or the blog post written by Ben Neals, because he wrote some the educational stuff. So you can have it on the link in the slide. Okay, so the first stuff we're going to learn about the introduction of the heritability, and we are overviewing the two different models. So first one is the Fisher model and the Falconer's model. Okay, so Fisher's model, as we learned in the previous session, he's the first person to introduce the concept of the polygenic model, and we called it infinitesimal model, and it proposed in 1918, right? So what he did is he tried to unify the concept between the Mendelian genetics and the continuous trait proposed by the biometrician, right? And then he proposed the concept of the polygenic inheritance, and that is explaining the quantitative traits in human population and also the natural population. So the things we can think about in a polygenic model as the factors, we call the additive and dominant factors, but also, I mean, there are other stuff like interactions or environmental factors too. But anyhow, in polygenic models, we have the different parts of the genetic or non-genetic factors involved in that and explaining the trait variance in a population. And also Fisher proposed the resemblance between the relatives due to their genetic covariance, right? So if you are from father, if you're from mother, you are more likely be your parents, but compared to cousins or second cousins or third cousins, right? So that is the sort of the familial distance related to your familial resemblance of your traits, right? And we learned about the Fisher, but he resolved the conflicting views between the biometrician and Mendelians, right? Okay, so in his Fisher's model, he tried to include all different parts of the multiple factors explaining the phenotypic variance, right? So phenotypic variance referred to how different heights in a population, right? So what is the distribution and how much they are differed by people in our population. And trying to explain that variance, Fisher tried to put different, the factors explaining that situation. So the two things about that, one is the genetic factors and non-genetic factors, right? And the genetic factors, including the three categories. The first one is additive, which are a little contribution to the phenotype. So it has to contribute a large number. And also another factor is called dominance, which is like, Mendelian monogenic situation. So one allele or one factors can drive everything in that individual, right? Or the situation like an epistasis, which is the interactions between the additive factors or additive and dominant factors. So this is the interaction between the genetic factors. And also he tried to explain the situation like, non-genetic factors can involved in the phenotypic variance, because as we know that the environmental factors can modulate your trait values, right? So how many times you eat good things during your life, how long you sleep during your, developmental age. So all of these things can be on the environmental factors. So, and those things are also involved in your phenotypic variance as well. So in his polygenic model, all of these things included, and we can easily formulate like this formula, which is the phenotypic variance refer P equal the variance from the genetic factors, variance from the environmental factors, plus the variance from interaction between genetic factors and environmental factors. So that was sort of the prototype Fisher proposed in early days, so about 100 years ago. And the people start think about how we formulate or how we articulate those polygenic models in our real genetic data set. Okay. And Fisher's polygenic model, it's based on the multiple compartments. So we can say that this is like a way to partitioning the variance, right? So if you want to explain the trait variance, then you can think about the multiple compartments. So in the sense we call it partition, right? So partition means that, I mean, if you are interested in computers or whatever the computer system, then some people wants to do the disk partition, because you split your hard disk into different positions. So partition means that you put it into the different parts, right? So as we describe in that way, when we think about the trait variance, the Fisher partition each factors into the different explanation, right? So partitioning variance means that the variance from the genetic factors or environmental factors. So this partitioning variance introduced the concept of the heritability, right? So heritability is a concept that summarizes how much the variation in a trait is due to the variation in genetic factors. So in other words, how much that traits or phenotypes can be explained by genetic factors or genotypes, right? So heritability is the way we can measure how much genetic factors can explain the trait, right? So sort of that the meaning. And in that sense, another important term called genetic architecture. So genetic architecture of a certain trait means that this is the composition of various genetic factors upon a phenotype, including the additive, dominance, and epistasis. And also this concept is further detailed by the type of genetic variance. So if you are explaining the situation like, what is the genetic architecture of type 2 diabetes? Or what is the genetic architecture of autism? Or what is the genetic architecture of schizophrenia, right? Even though they are the same disorder, but they are differently explained by different genetic factors and environmental factors, right? So the notion is that we are measuring which genetic factors or environmental factors can change the phenotypic variance or can explain the phenotypic variance. And that is the way why we are doing a heritability study. And also in that way, we are considering the concept of the genetic architecture because even though we have the same disorder, but can be explained by different regions of the genetic factors, right? So when you think about the human disease or human genetics, you have to have the sense that the same disorders can be explained by different regions of the genetic underpinnings. Okay, so in other words, genetics in these days is to identify genetic factors associated with trait or disease, but also study the contribution of genetic factor, right? So this is quite critical for our students because probably you're going to be engaged in the field of biomedical in some days or pharmaceutical or whatever the medical situation. In anyhow, if you are in the clinical context, you are forced to think about the genetic architecture of the disease because the genetic architecture is the core term you have to understand and everyone wants to study in these days, right? The reason is that, if you see two patients, they are all coming to clinic because of the type 2 diabetes. And one person has the problem of additive factors in their genome, in her or his genome. And the other patient is affected by dominant factors, right? So that is quite interesting situation because they are all diagnosed for type 2 diabetes, but they have different regions of the genetic changes, right? So one is affected by additive factors and the other is affected by dominance factors. Then your question is that, can we try to, give a same, drug for them, right? Or we have to make up different plan for the different, drug medication, right? So additive person probably, other, drug A is more effective, or the other one is more, interacted with other drug portion, right? So all of these things has to be considered in the way, and also think about different situation to, whether they have the different phenotypic values or whether they have different clinical symptoms, right? So all of things coming to the questions of what is the genetic architecture of the disease, right? Because we are on the same diagnosis, but have the different levels of symptoms and phenotypes. So that's why people want to do that. And also this is linked to the idea of why people want to do sort of the large scale data set or whatever the wearable device or whatever different notions of the biomedical research. The reason is that people want to see the different, phenotypic values or trait values within the same, diagnostic criteria, right? Because of the different genetic architecture, right? That is the reason for that. Okay, so based on this features model, people start think about, how we continue to improve this idea into our genetics model. And one of the pioneers at early days was the Douglas Falconer. He's actually working in the behavior research rather than the genetics. So he's working on the depression study using mice in 1950. And while he studied the mice behavior of depression, Falconer suddenly noticed that the phenotypes in an experimental model show normal distribution, right? So even if he studied the caged mouse in a, whatever the isolated situation, even though that the behaviors of these, the mice is actually having some distributions in terms of their phenotype, right? And this distribution is heritable and also can be, subject to, measurement, how much the genetics is working in that in the situation. So Falconer introduced his formula for calculating the heritability, and that is the sort of the first model for the quantitative traits in genetics history. Okay, so what he proposed at the time was he thought that the twin studies is the easiest way we can measure the genetic contribution of traits in the family, right? The reason is that if you look at the twins, we can split into the monozygotic twins and dizygotic twins, and monozygotic twins has to have the identical genetic material, right? So they basically have the same genetic makeup from the between the twins, right? But dizygotic twins has the only 50% similarity because they don't share the germ cells, right? They only share the environmental portions in the family, right? So Falconer's idea in early days was measuring the phenotypic correlation, concordance within the monozygotic twins, and then measuring the same values from dizygotic twins, right? So he measured let's say the height is the concordant in the monozygotic twins, and then the level of that concordance from the zygotic twins, and measuring the differences between these two types of twins. Then he can easily explain that, okay, so these differences is the part of the genetic makeup we can explain for that trait, right? So let's say monozygotic twins have the concordance of 0.86, let's say from height, and dizygotic twins has the concordance of the 0.6. Then you have the 26% differences between the mono and di, and you can easily times 2, then explain that the heritability is the 52%. The reason why we times 2 is the monozygotic twins has at least the half of the genome share between these two. So we can times up 2, so make up the entire 100% scale for the genetic makeup, right? So that was the early version of measuring the heritability proposed by Falconer at the time. And of course, that is more, improved over the time using the different aspects of the phenotypic values in our population. So thinking about that notion, we had to learn about some, the key terminology describing the phenotypes in our population. So the first, the core idea is the normal distribution, right? So normal distribution is the easiest way we can explain the heritable traits in our population. And normal distribution is the one of the type of distribution. So it definitely implicates the continuous perspective of the traits in our population. And also using that the normal, the characteristic, we can easily measure the mean value of the trait in a population, and also the deviation that each individual has in a population, right? So the two concepts of the mean and variance is explaining the characteristics inherent in our traits. Okay, so what is mean? And I think this is the high school math for you. And this is the sum of all the measurements divided by the number of the measurements, right? So in here, a measurement can be like height, weight, or arm size, or arm length, or whatever mental activities or physical activities, or your metabolite, the performance or something, right? And then variance is the average square deviation of the observation from the mean. So variance is explain the situation, how deviate individuals from the the mean value from the population, right? So these two things is describing the quantitative aspects of the traits in our population. And this is explaining the sort of the genetic levels of each individual having in their genome, right? So distribution of the trait in a population means that the proportion of the individuals that have each of the possible phenotypes, right? So if you want to know about the, how high people in how tall people in our population or how short in our population. So that case, you use the normal distribution and put it into that measurement and think about the mean and variance, right? But you can also imply that notion to other activities we can measure in different clinical studies or different whatever the natural experiments, right? I mean, here I explained the sort of measuring the deviation using the normal population, because the normal distribution is literally means the normalized value from your population. So that means that we use that symmetry perspective in a distribution. Then we can easily explain that how individual is the deviate from the population mean, right? So that means that if you see some individual have the the phenotypic value have 3 standard deviation from the mean, then you can easily say that that individual has much severe or much stronger phenotype than the individuals have like a 2 standard deviation or 1 standard deviation, because we put all of these phenotypic values in a scales of the normal distribution. Then you can easily interpret these individuals has the strong phenotype or mild phenotype or something, right? And also this deviation is underpinned by the genetic factors as we launch in the polygenic model, right? Because the reason why that individual on this extreme or on this 3 times standard deviation, the reason is that these individuals probably have much higher, the genetic variance from the, compared to other individuals, right? So all of this distribution is the same notion of how much that individual has that the genetic factors or genetic underpinnings, right? So normal distribution is one of the easiest way we can easily explain the the phenotypic deviation and also genotype deviation. But then also you also have to keep in mind that the normal distribution doesn't tell anything about the inheritance, because this is only merely describe the phenotypic distributions of the individuals in a population, right? So if you want to describe the inheritance situation and in the continuous trait, then you have to you have to add one more layers onto this normal distribution. So we'll cover that issue in the next slide. But also, I want to emphasize one more thing is that the phenotypic values we can describe from our population, let's say human, it's not always following the normal distribution. So distribution can be, other types like, Poisson distribution, binomial, the negative binomial distributions or blah, blah, blah. So here is the normal distribution is one of the examples we can, put it into sort of, I would say conventional situation, but we have the other types of the distribution in our traits. So you have to keep in mind that. So if you want to study something in future, or if you are in the situation for the diagnosis or something, then you have to think about traits of or the clinical conditions of your interest following the which distribution, right? So that is the one note for that. And also, if you are really serious about the knowing about the distribution, and also think about the statistical perspective in genetics, then you can read about the differences between the population and samples, and sample variance, sample distribution, and population distribution are different, and that is the one of the classic way we can describing the distributions in statistics. So that will be like the second year topic. So you can search that things and think about how these two things are different in real world. So that's the one issue. And okay, so let's continue discuss about the distribution of the traits does not imply the inheritance, and that has to be related to the concept of familial resemblance and covariance, right? Okay, so genetics wants to do identify the resemblance between family members. So that is our aim. So how can we do that? So we used covariance, which is the mathematical concept. That is the measuring of the joint variability of two variables. So here it means that two random variables means the trait, right? So we want to compare the similarity between two populations, right? So two population means that the first one on here, X, is probably the parents' values, and Y is the child values, right? We want to compare the resemblance between the family members. Or you can put the child and you can put the grandparents, or you can put the child and you put the whatever the second relatives or third relatives, so the familiar relationship. Then you measure whether these two populations, they are similar or not. In that case, what we know is we know that the trait is following the normal distribution, and then we're using that we're using the concept that these two distributions are the similar or not. That case, we use the concept called covariance, right? So the covariance is the measuring the deviation between these two populations. So let's say you have this dot here, right? So this dot is the pair of the family A, let's say. So family A, so this is the father's height, and this is the child height, right? So from one family, and the other family has this pair and this pair. So you basically measuring the deviation between these family members' values to the mean, right? Or this value from the child to the mean, right? So the covariance is the measuring the deviation between the two populations. So one is the deviation from the X population, which is the parents' values, and the other deviation measured from the child values. So measuring the each child heights to the mean of the child height, right? And the covariance can be explained the relationship between these two populations. So here has these two populations has the zero covariance, which means that the two populations doesn't really have the similarity between, I mean, in terms of the trait, right? And this situation has the plus covariance, and that means that these two populations has the resemblance in terms of the trait values, right? And this case is the anti-similarity, so discordance, right? So it's the reverse relationship between these two populations. So covariance is using the plus or minus values to explain the direction and they explain the similarity between these two populations. So that is one of the easiest way we can measure the resemblance between family members, right? So in other words, we can simply put on that context. So if you look at the mid-offspring beak depth, so which means that the length of the beak on the birds, right? So you're measuring the biological mid-parent's beak depth and the child values. And also here is the foster's mid-parent beak depth, which is the non-biological parents, right? So clearly we can, assume that this is genetics and this is the inheritance situation, right? So there has no, the direction on covariance. So we can easily say, the big depth is not, the big depth is the inheritance situation, because we see the positive covariance values on the biological mid-parents to their offspring, right? So this is the easiest way we can measure whether this trait is the heritable or not, right? So the covariance is one of the easiest way we can measure the situation. So why we care about the resemblance between the family members, right? So the reason is that the family members is the easiest way we can think about the expectation, right? So as I explained about the monozygotic twins, or in other words, identical twins here, right? That case, we have the 100% the identical genetic makeup, right? So they have the same DNA sequence between these two. Then if you look at the fraternal twins, which is the dizygotic twins, that case we have the 50%, the sharing the DNA between these twins, right? So this 50% can be a expectation you can measure from the phenotypic values, right? Or if you look at the parental-child relationship, or if you look at the full siblings, then it is also the 50% the similarity, but slightly offset from this expectation. But roughly we can say that 50%. And also, if you add one more familial relationship, we say distance, then the expectation reduced by half, right? So grandparents is 25%. And aunt, uncles, niece also 25%, half siblings 25%, so and so on, right? So this is the easiest way we can expect from the family member relationship. Okay, so using the expectation, then you see the covariance value and you see the phenotypic values and whether that trait is the heritable or not. So this is sort of the the criteria you can think about that the inheritance, right? Okay, and also one interesting note from the Wilhelm Johansen in 1903, and this is the measurement of the parent seed weight and the offspring seed weight, right? And you can clearly see the similarity between the parents and offspring in terms of the seed weight, right? And there is a, good correlation good similarity between these two, right? So that is the good thing. And why I put this one in here is that I found this quite interesting because the Wilhelm Johansen is one of the strong, Mendelian people, right? So Johansen is the, strong supporter for the Mendelian, inheritance. And he obviously did experiments to prove, Mendelian genetics is, ruled out, right? But then in his data set, we visit the data set and plot into like this graph. Then you see the variance within that family, right? Within the parents and offspring relationship, right? At the time, Johansen didn't really notice that, this variance is explained by environmental factors, right? He just explained that is probably the offset by measurement. But actually, nowadays, we know that the sort of environmental factors are also involved in the phenotypic variance. So, I mean, we can clearly claim these examples for that situation, right? So it was quite fun things why with the Lynch's textbook that is appeared in here. Okay, so back to resemblance situations. So when we measure mathematically for the resemblance, we use the slope concept. Okay, so slope is the how the steep of the lines in a data point. So in this case, we can easily compute the slope from the covariance divided by the variance we measure from the X population, and that is indicating the how similar the traits between the familial relationship, right? So as we expected from the familial relationship we discussed in here, monozygotic twins has the 100% correlation between these two, right? So the slope has to be close to 1, right? And that is the steep slope appeared in here. And then we have the first siblings and parental offsprings. The reason is that these two share the 50% of the genetic makeup in their familial relationship, right? So this is the discussion we get from here, right? But interestingly, forceps are known to have slightly higher slope than parent and offspring. The reason is that the relationship between parents and offspring, they don't share the additive factors, right? Oh, sorry. They don't share the dominant factors, right? The reason is that the parents and offspring relationship only explained by additive situation, right? So parents can have the same additive genotype as offspring in their genome, but they don't have the same homozygous genotype as offspring, right? The reason is that parents' genotype is made by grandfather and grandmother, right? And parents only transmit one of alleles from grandmom or granddad, right, to offspring, right? And offspring's homozygous genotype has to have the same genotype from the genotype makeup from his father and mother, right? So if the homozygous genotype happen in the offspring, the homozygous genotype never going to have the same as the father or mother's homozygous genotype, right? Because parent and offspring only share the one strand of the chromosome. This is the important things. But if you look at the full siblings, that is the different situation. The first siblings can have the same homozygous genotype because they share the parent, right? So that's why the slope of the genetic similarity in forceps is slightly higher than parents' offspring, right? So it's a little bit slightly than 50%, okay? And the hopseps, as expected, that is the 25% of the expectation. Okay, so that is the why we have the different, the slope steepness. And there is one thing. And also, when we explain this resemblance, we use slope. We don't use the correlation coefficient. So I accidentally mentioned that the correlated between these two families, but it actually the similarity or resemblance rather than correlated, right? Correlation and resemblance is slightly different concept in terms of the mathematics. The reason is that correlation coefficient is the measuring in a different ways compared to slope using the covariance and variance. And if you have the fuzzy distribution in in your data set, correlations is always can happen as a higher value, regardless of the covariance situation. So we are a bit cautious on to using the correlation coefficient in measuring the resemblance. So we take the more, the earlier versions of the math onto measuring the resemblance, which is the slope in here. So if you want to know more about that issue, you can go to this link. Okay, so next topic is the heritability. So heritability, then we can discuss about different, the parameters in here. So phenotypic similarity in family depends on the genetic relationship and also traits, right? So total phenotypic variance for character, we noted as a VP or the sigma P, the squares. And this is the functions of the phenotypic variance is equal to genotypic variance and environmental variance, right? And this means that the combined effects of the genotype and also environmental factors, the variance of genotype and the environment, right? And genotypic variance is explained by the differences by different genotypes. So which genotypes you have, then that will make changes, or they will make differences between individuals, right? And also the same logic for the environmental variance as well. So think about the situation that we have the millions of the genotypes or alleles in our genome, then which combinations of allele or genotypes in your genome that will make a differences in our traits, right? So that is the way why we think about the variance of genotypes, right? Okay, so genetic variance can be divided by the different genetic factors, as we discussed in the Fisher's model. So the first one is additive genetic factors and dominant genetic factors and epistemic genetic factors. So additive genetic factors, we noted VA, and dominance is the VD, and epistatistics is the VI, right? And this is obviously, the variation due to the additive effect of allele, or variance due to the dominance relationship, or variance due to the interactions among loci, right? Okay, so the first concept you have to understand is the differences between the dominance and additive variance, right? So dominance variance is due to the dominance deviation, which is described the extent to which heterozygotes are not exactly intermediate between the homozygotes. And this is the same concept we define in Fisher's single locus model we discussed in the section 2 and 2, right? And additive genetic variance is responsible for the resemblance between the parents and offspring. That is the one clear example for that. And additive genetic variance is the basis for the response to selection, right? So we can more detailed with the examples. Okay, so this is the plot we call the liability threshold model, and this is the classic the plot for we describe the phenotypes in a disease, right? So if you study biomedical stuffs, we always dealing with this the normal distribution plot for describing the phenotypic values in a population, right? And that is actually first proposed by Carl Pearson and Lee about centuries ago, right? So this is the same normal distribution plot, and each individual is sitting on somewhere in here, right? So let's think about this is the height, right? And maybe for the Korean population height, and let's say the female trait, right? Female height. Then the average of the, I don't know how tall the Korean women's, but let's say 170, and that is the average. And some, the person probably have like a 220s or 210 centimeter. or some people has like 70 centimeters or 80 centimeters, but they are unlikely happening in the population. So most people have around this range, right? Then we can say people are short or tall, right? And the reason why we have the less people on here is the reason is that the individuals in that area have the higher selective pressure. So they are unlikely to be mated in a population. That is bit, sad to say, but that happen in an evolutionary perspective, right? So let's say if you are sitting on here, so you're a Korean woman and you're, the height is about 175 and you are sitting in here, then the reason you have that height value, the reason is that, many of your additive factors is working in your genome, then that drives to you make the height on this value, right? Or if you have one strong dominant, alleles in your genome, then you have 120 centimeters for your height. Or that dominant alleles can, have large effect, but not that large as much as this allele, then you have like, 195 centimeters and you're sitting on here, right? So if you think about the traits, not just the height, but other things like, autism, schizophrenia, or, type 2 diabetes or metabolic traits, glucose uptake or something, all of these traits are explained by how, alleles combined and result in the phenotypic values. So this is all different composition of that. So I'll give you some examples of some extreme case. So the cases of the achondroplasia and Marfan syndrome. So achondroplasia is the case where the person has the extremely short stature, height. So they have normally like 70 centimeters or 80 centimeters. The reason why they have the short statures, the reason is that 98% of the achondroplasia cases, they have the mutation on FGFR3 gene, and they have the mutation on this position, right? And because of this one allele change, one DNA sequence change, make their height quite short, right? And they bring that dominant allele, bring that to here, right? Or the other case, like a Marfan syndrome, which is happening the mutation on FBN1. And if you have the missense mutation in this gene, then your height is around 195 or 200 or 210, right? And that case, Marfan syndrome, is less selective pressure than achondroplasia. So 75% of the cases they can marry, they can mate, they can have baby, they inherit their mutation to the next generation. So that can explain by 75%. And 25% of cases, they are affected by de novo mutation, which is newly occur in sperms or eggs. And then mutation happen in the child, right? So some of cases have the tall parents, but some of cases have the normal height range parents, but suddenly the child can have that very high height stature. The reason I explain this is that these two cases are explained by the dominant factors onto the height value, right? But most of the people in our population, height is explained by additive factors or the combinations of the additive factors plus the effect size of dominant factors, right? So this is all mixed up of these things, right? So you have to understand about what is the differences between the additive factors and dominant factors. Then the question is, how many additive alleles affecting or influence on the height value, right? We don't know yet, but genetic research studies so far, we discover 300 alleles affecting on human height. and that is explaining some portion of the population. But as the research is going on, then we see more alleles and more alleles, then ultimately we can predict the offspring height value using the genotype information. That is the one of the notion people wants to study that. But of course, the height is not really the people wants to know about the next generation. People wants to know about the disease risk, type 2 diabetes, the chance of having depression, chance to having the glucose problem or something. So people want to study that first, right? So we are still studying how much additive factors or dominant factors are affecting on our trait, right? So in that context, we are studying the different genetic architecture. The reason is that even though it's all same height, but the height can be explained, some cases explained by additive factors, some cases explained by dominant factors, right? Or some people explain by environmental factors, right? But studying environmental factors is slightly trickier than the studying genotype information, right? So you will be quite surprised that you probably learned about the genetic disorder in high school, like if you have the problem in this gene, then that person has that disease. But as we study more and more genetic studies, we see many exceptions, right? As we see the diversity in a population, when we study more and more, we see different parts of the genetic architecture. So that's why I keep explaining polygenic model is the critical thing, genetic architecture is the critical things, because this is more modern genetics we are dealing in 2021, right? And one TMI. So FBN1 is the gene working on the Marfan syndrome, which makes you tall, right? But interestingly, some mutation on the same gene, FBN1, makes you quite short, right? That is the happening in the recent study in Nature. If you look at some Peruvian people, so Peruvian is the people living in the Peru in South America. And if you look at some of the people near the coast, they are quite shorter than the people in the Peru on the mountain, right? So people are interested in why geographical differences make different population height value. And what they found is the one missense mutation in the same gene, right? In this FBN1 gene, right? And that makes Peruvian short, right? It's interesting because some mutations makes you super tall, but some mutations makes you short, right? Even in the same gene, right? So that is another, the interesting topics we also dealing with the genetic architecture, right? Okay, so if you want to know about the genetic architecture, you also have to think about the IBD, which is the identity by descendant, and also the genetic relationship. So this is again the same explanation as the familial resemblance we discussed on here. So more proper, the genetic terminology we call the IBD. And the easiest and also basic principle is explained by additive factors, right? So additive relationship between X and Y population, which is the, let's say, parent and offspring, right? And this additive genetic relationship, a portion of the alleles that have the X and Y have in common. So if you look at certain allele, then you count that whether the allele is commonly seen in the father and child, right? Or mother and child, right? So if you, in the context, you can easily explain that the parent and offspring has the 50% expectation in terms of the same allele composition in their genome. The reason is that, as I explained, the parents, let's say father, transmit one strand of the chromosome to offspring. So only 50% is transmission happening the next generation. And two full siblings has the 50% too, if you only care about the additive effect, additive composition. And two half siblings also the reduced by half, which is the 25%. Okay, so if you look at the phenotypic covariance, you have to think about the different genetic factors. So one is we are discussed the additive genetic variance just before the slide. And another case is the dominant genetic variance and the variance due to the shared environmental factors, right? Okay, so let's detail on that. So if you look at the monozygotic twins, and they perfectly have the same genotype between these two, same DNA between these two twins, right? So they have the identical additive genetic variance, right? And they have the identical dominant genetic variance because they have the same DNA. And then they grown in the same environmental situation, right? We assume that they grown in the same environmental situation. But of course, there are some exceptions on this case, like, Romanian orphanages. So if you are interested in that, the history of the Eastern European after the, Second War, then Romanian dictator, they, set out all, the twins into the different, the country. I mean, if you are a fan of the Netflix drama 멜로가 체질, then you see the scene is appeared in the very last episode because I'm a big fan. But anyway, so the Sogu mentioned that the Romanian documentary or something, but there is actually the one exception for the the environmental the different environmental of the monozygotic twins. Sorry, I mean I'm too excited about the drama, but anyway, okay. So that case, well, the monozygotic twins has all the same compartment, and the parent and offspring, they only have the same additive genetic variance, but only 50%, because the father transmits half of that, right? And the full siblings has the 50% additive, the same proportion, because they share the 50, and the 25 chance of the dominance. The reason is that they have the 25 chance of the homozygous genotype. Think about the Mendelian ratio, and that is they can have the same homozygous genotype from the parent, right? And this is one of the things people normally confused about, why they have the same homozygous genotype. So draw the relationship from the grandparents to parent and child, then draw the chromosomes and alleles, then think about the probability in each situation, then you will know that the homozygous genotypes can be only same in the full siblings, not in the parent and offspring relationship. And they grow up in the same environment, right? So parent and offspring do not grow up in the same environment, because parents, grown up in the different place because they are different ages, right? So they are different, right? But full sibs, they are normally growing in the same environment, right? And also grandparent and child has the 25% relationship between two. So that's why this slope has that steepness, and the full sibs and parent offspring differences between these two as well. Okay. Okay, so heritability is as I mentioned, that this is the measurement of how much the genetic factors is explaining the phenotypic differences. Then I can easily recap that for Connor's formula using the twins following this formula. And then heritability has the two methods to measure the heritability. So the first way is the narrow sense heritability. We normally call small h2. And this is the measuring the genetic variance only using the additive genetic factors, right? And the broad sense heritability is the measuring all of the genetic variance from the population. So using the dominance, additive, and interactions, right? So this is the complete versions of the heritability measurement, and we call the large H on here, right? So why we are distinguish between the narrow sense and broad sense heritability. The reason is that in real world, getting the samples for the research, we are easy to get a relationship for the additive situation. So if you want to measuring the dominance or epistatistics, then you have to get the cases for the forceps, right? And getting the information from the forceps is not always available. So people always take the easiest way and more realistic experimental design. So the first way, we developed the narrow sense heritability and using the additive factors on that. And more recently, some studies tried to do some broad sense heritability measurements, and they tried to measure the contribution of dominance factors, but also measuring all of these factors is limited in terms of the sample recruitment. Okay, so if you think about the heritability, and if you, naturally be curious about, which traits we see in a life, daily life is high heritability, right? So if you look at the hair colors, that's probably quite high heritability because you have the black hair, your, child has black hair too. It's unlikely to have the red hairs or whatever the other colors, right? Blonde hairs. So hair colors is definitely a higher value. This is again, they have more dominant and also more sorts of the dominant-ish genetic architecture. So like hair colors or skin colors or eye colors, they have more higher heritability. And also height has higher heritability too. Musical ability or musical taste has less, heritability. We can instinctively think about the situation as well. So think about, the phenotypes or some activities you see in the daily life and have a question whether that is heritable or not, then you can search that things on the, whether that issue is done or not. So I'm not sure whether we can cover that study in this semester, but there's extremely fascinating study published in 2 years ago about the homosexuality, right? And that's appeared in Science in 2 years ago. And people debate about that study, whatever they think, and no one really understand about the genetics at all. But Korean media, they saying blah, 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 whatever they want. Interestingly, none of the biology, the professors can comment on that. That's quite, pathetic because, we are not the, polygenic trained generation, right? So, but again, the notion of the modern genetics is going on that direction, and people studying, the genetic architecture of the polygenic aspect of many different phenotypes, and that's definitely put it in the public health relation or other, society issues too. So, keep in mind that all of these things are heavily relevant for your future career. So, some people might find bored because, this is totally new things. You probably felt awkward in the context of Mendelian genetics, but all of this, the story is brought to you in the context of the modern genetics. Okay, so one last very short explanation about the heritability, how we measure heritability in these days, in 2021. So we are not using the familiar relationship anymore because the technology advanced and also the methodology advanced over the time. So when Falconer's day around 1970s or 1990s, we heavily rely on the familiar relationship to measure the heritability. And at the time, we didn't have the genotype information from the population. So we can only measure the phenotypic variance, and we can only measure the familial relationship, and we're measuring the heritability. We calculate the heritability using the two types of information. But nowadays, we have the genotype information because we have the advanced genotype technology introduced in late 2000. Then people, make a genotype information, and we're measuring the heritability based on the SNPs, right? So SNPs in this context is the same meaning of the alleles or genotype, and that is the meaning of the familial relationship, right? So how much the same SNP have in your genome, in the population's genome, then how this SNP similarity, combination similarity as the similar to the phenotypic values, right? So you set up the phenotypic measurement between the case and controls. So let's say controls and type 2 diabetes patients. Then you measure, then you're using the SNP information and how much people share the SNP values. And then we measure whether type 2 diabetes is how much heritability in our genome, right? So this case, SNP can be used as the instrument of measuring the familial resemblance. So sort of the equal instrument for that. Okay, so if you're really serious on this issue, you can read this paper, but this is quite mathematics, but this is one of the famous the study in the world because this is the first paper measuring the SNP heritability in human history, and that's written by Korean. Yeah, I mean, you can be proud, Hong Lee, right? And I mean, I have lots to say about the story of the paper, but again, this is also quite new to our generation. So I mean, the learning materials I introduced today is you're not going to have the same things in other class. So please have and also expand your thoughts and how the field is moving on in these days, right? Okay, so quick summary. So heritability estimates the degree of variation in a phenotypic traits in a population due to genetic variance between individuals in the population, right? So that is the key sentence for that. And also we learned about the concept of the quantitative trait and also the properties of the normal distribution, how we represent the traits in we observed from the population. And then also the genetic similarity due to the familial resemblance, familial relationship, and that is the way of we measuring the heritability based on the covariance concept, and also the familial relationship. We learned about that. So think about the core concept we dealt in here, and probably one of the most hardest thing is why forceps has the dominance effect. And that is why there is the absence in parents and offspring relationship. Okay, so that's everything we covered today. And I have some further readings on today's topic. So we have Heritability 101 and 201 post by Ben Neal, and there will be interesting stuff for your learning. And also, we have quite classic review of the heritability, and I think that is probably the textbook chapter equivalent material, and that is written by Peter Bishop. Peter Bishop is the person, the first developed this area, and he's like a demigod, and in a quantitative genetics in these days. So this is inspiring review, and you can have some basic sense on that. And also, what is this? Okay, so this is how Douglas Falconer found the heritability and how he got into the field in the quantitative genetics. And this is sort of the historical events on that. So if you are, seriously want to know that, and that will be interesting review. And if you are super curious about the quantitative genetics, probably for your career, Then Falconer's textbook is one of the Bible in a quantitative genetics area. And also there is another book called Statistical Genetics. It's written by Benjamin Neal. I don't use this book for the undergraduate course. I only dealt in the graduate graduate course. But this is amazing book for the the large scale genomics. And if you are really serious on the I want to do this in my career, then this is the starting point for your textbook, because I mean you have lots of time to read the materials, and this is good. And Ben Neal is probably the next demigod in the field. He's an amazing guy, and I think he wrote this book when he was 21 or 22 something, when he was visit Oxford for his summer vacation, probably, my guess. He's quite young guy. But this is great book, and it's covering all of the classic quantitative stuff, and also linked to the modern genomic research as well. And Ben Neal is obviously the top leading scientist in the genetics in these days. So I mean that will be quite inspiring for your learning. And I'm not sure our university has this book or not, but yeah, you can borrow from the Amazon yourself. Okay, so that's everything. And if you have questions, leave the comments or questions on the Slack. And also if you have anything unclear during your exam preparation, use Slack for that. And also some of students are quite shy or isolated, so not to give a question. But I mean, I wouldn't really recommend to do that. So always welcome to the questions. And also some people are quite fear about, this class is the English, whatever, genetics things. So quite, fear on the exam preparation. But one thing I can tell you is that, I mean, the exam result in previous years is not, related to how much you do the English, right? So this is the matter of how you understand the genetic concept, right? I mean, I can see the variables of the international students to the exam results. So I mean, if you are confident on the English, don't be too confident on that. So so prepare your stuff, and also be on Slack and prepare your exam. So we're going to follow up the exam preparation next Monday and Q&A session, and hopefully you enjoy the topics, and see you next time.